This is the fourth section of our anaerobe lecture, and we're going to continue our discussion on some of the Clostridium species, and we're going to start by talking about another Clostridium, and this is Clostridium difficile. Here is a gram stain of Clostridium difficile, and it is a gram-positive rod. Now, we already mentioned that the Clostridia organisms can sometimes over-decolorize. So you can see in this gram stain, this is a classic Clostridium gram stain, where there are portions of the cells that are staining a nice dark purple, but then there are portions of the bacterial cell that are staining more of a pink. So it is gram-positive, but it can be over-decolorized. And you could see that clear area inside the bacillus is the spore. So Clostridium difficile is associated with the disease pseudomembranous enterocolitis. And usually, an individual will get pseudomembranous enterocolitis caused by Clostridium difficile after they have been on antimicrobial therapy, and this is considered an iatrogenic infection. So many individuals carry low numbers of Clostridium difficile in their gastrointestinal tract. And our normal bowel flora, like our E. coli, keep the numbers of some of these organisms that can become pathogenic, keep them at low numbers. Now, if you're taking antimicrobial therapy and some of your normal GI flora gets reduced because of the antibiotics, it can allow an organism that's present but kept in check to overgrow. So you can pick up Clostridium difficile in hospitals. They're common in hospital environments. And again, there are 3% of healthy adults in the population have Clostridium difficile as part of their normal intestinal flora. So you can allow that normal flora to overgrow, and you can end up with pseudomembranous enterocolitis. Now, Clostridium difficile has the ability to produce two different toxins. Toxin A, which is an enterotoxin that causes diarrhea, and toxin B, which is a cytotoxin that causes cellular damage. And here is the typical pseudomembranous enterocolitis that might be caused by Clostridium difficile, where you get inflamed intestines and some ulcerations, and from what I've heard, this is an incredibly painful infection. We're going to now talk about the epidemiology of the Clostridium species. Now, some of these organisms can be intestinal flora. You can find sometimes Clostridium perfringens, tetany, and more commonly Clostridium difficile as intestinal flora. A lot of these Clostridium organisms can be found in the environment, in soils. They can be found in various animal species, and transmission, depending on the organism, is usually through traumatic wounds, ingestion, or endogenous. So you get an infection with your own flora. Prevention and treatment for tetanus, there is a vaccine. It's part of the DPT, diphtheria, pertussis, tetanus vaccine. You can prevent botulism by proper home canning of food and being careful about not eating food that could potentially be contaminated with Clostridium botulinum that might be containing a preformed toxin. Don't give honey to any children less than 1 year of age, and they usually tell new mothers, remind new mothers not to give your children honey until after they're 1. There are some treatments. Clostridium tetany and Clostridium botulinum do have antitoxins, but keep in mind they're not that effective once the toxin is bound. For Clostridium perfringens, you can debride the wounds, try to get rid of the area that has toxin being produced. Supportive therapy, such as for botulism, respiratory assistance. Remember the flaccid paralysis where people can't breathe, you would have to give them respiratory assistance, as well as antibiotics. To diagnose Clostridium species, we already mentioned, and you saw some pictures, these are gram-positive rods. They easily decolorize and can appear gram-negative. Clostridium species are sporogynous. They are spore formers, but keep in mind, when you gram stain a clinical specimen, you commonly do not see the spore. They just look like gram-positive rods, and they can look like a mixture of gram-positive and gram-negative rods because a lot of the organisms will over-decolorize. Now, if you do special spore stains, you will see the spores, and the spore placement can be helpful for identification. So on the top left, Clostridium tetani, where you see the terminal spores, those tennis racket shaped organisms. And on the bottom right is a different Clostridium species, which is Clostridium sordellii, and that organism has subterminal spores, so a spore near the end but not directly at the end of the bacillus. Now you always want to culture your anaerobes in an anaerobic atmosphere. You are going to use primary isolation media that contains blood. Commonly it's a Brucella blood agar plate, and you always want to incubate for at least 48 hours before you open up your anaerobic jar and check your plates. You can do some tests for presumptive identification. So we mentioned some of the selective media. There are lecithinase anaerobic egg yolk agar media, and that is very good for presumptive identification of Clostridium perfringens, which produces the enzyme lecithinase and will make an opacity around the colonies when they're streaked on the anaerobic egg yolk agar. Clostridium botulinum produces the enzyme lipase, so you can use special media and you'll see an iridescent sheen around Clostridium botulinum colonies, and you can look for the you can presumptively identify Clostridium perfringens using the Nagler reaction. So what the Nagler reaction is it's an inhibition of the enzyme lecithinase that's produced by Clostridium perfringens using an antitoxin type A. So on the top left, you could see the lecithinase reaction, that zone of opacity around the colonies growing on the anaerobic egg yolk agar. And on the bottom right, you could see also on egg yolk agar the lipase reaction, where you see a iridescence around the colonies. Again, here's your egg yolk agar with Clostridium perfringens showing lecithinase activity. Now here is a actual picture of a plate and a schematic of the Nagler reaction. So what you would have is you have your egg yolk agar plate where you would be looking for your lecithinase production. So you are going to have your lines going across of your different Clostridium suspect species. On one half of your plate, you have the anti-alpha toxin, and on the other half of the egg yolk agar plate, you don't have any toxin. So on the side that doesn't have any antitoxin, if you have Clostridium perfringens, you're going to have a positive lecithinase on the egg yolk. You're going to have that opacity around the growth. On the left-hand side, if the antitoxin A inhibits the production of lecithinase, so you don't get that opacity around the colony growing on that side of the plate, that is an indication that you definitely do have Clostridium perfringens. So for Clostridium perfringens, you can look for lecithinase positive. You can look at that Nagler reaction where you inhibit lecithinase using that antitoxin A. On a blood-based agar, Clostridium perfringens forms a double zone of hemolysis. You can also do the reverse camp test where you will streak beta strep group B perpendicularly to your Clostridium perfringens and look for that enhanced zone of hemolysis. You can also do biochemical testing, which is your definitive identification method. So here is Clostridium perfringens growing on thioglycolate in thioglycolate broth, and you could see it's growing only at the bottom of the tube where the conditions are anaerobic, and on the right it's an anaerobic blood agar plate. And here on sheep blood tryptase soy agar plates, you could see Clostridium perfringens, and you could see a double zone of hemolysis where you have a very clear area in the center of beta hemolysis surrounded by an area of alpha hemolysis. And here again, that nice middle area on the top left of your beta hemolysis surrounded by a zone of alpha hemolysis, and it kind of looks like a bullseye. On the right is Clostridium difficile on another specialized selective anaerobic media. And here's a flow chart for the gram-positive anaerobic bacilli. So does it have a double zone of hemolysis on a blood agar plate? If it does, it's likely Clostridium perfringens. You would also see a boxcar-shaped bacillus. It would be lecithinase positive. It would be reverse camp test positive. You would see a subterminal spore, but remember in clinical samples, you're rarely going to actually see the spore. If it does not have a double zone of hemolysis, you're probably thinking of some other Clostridium species. So you definitely want to see if there's lecithinase production on your egg yolk agar. If there is no lecithinase production, you'd look for lipase. If there's lipase on egg yolk agar, you might be looking at a Clostridium sporogenes. If it's negative, you need to do some other biochemical tests. If it's positive for lecithinase, you want to look for lipase. If it's positive for lipase as well, it's a different species of Clostridium. And some of these other species of Clostridium very rarely or are never involved in infection, so we only talked about the ones that you would more commonly be identifying in the clinical setting. So you can go through this flow chart, but a lot of anaerobic infections are diagnosed based on the history. So if someone has a trauma, someone ingested something, the symptoms, and then the biochemical reactions. So we are now going to talk about the anaerobic gram-positive bacilli, but these are non-spore formers. So the Clostridium species form spores, and these next organisms do not form spores. So the genera that are gram-positive bacilli that don't form spores are Actinomyces, Bifidobacterium, Eubacterium, Mobiluncus, the anaerobic lactobacillus, and Propionibacterium. Here's a gram stain of Actinomyces. This is a gram-positive branching organism. We mentioned Actinomyces way back earlier in the semester when we talked about our gram-positive branching rods. And the last slide said, keep in mind, Actinomyces is an anaerobe. So although it is a branching somewhat filamentous positive rod, it's different from those in the fact that it is an anaerobic organism. Actinomyces is the cause of actinomycosis. which is more common an infection in the brain, the orofacial region, the pleuropulmonary region, as well as the genitals. It can cause a chronic granulomatous infection where you can, it'll start out as an infection in the sinus tract and the fistula in that area, and you can get a draining pus that contains sulfur granules. So on the top left, again, a gram stain of Actinomyces israelii. You can see the nice gram-positive filamentous rods. And on the bottom right is a gram stain of Mobiluncus, which is a gram-positive rod, but keep in mind these gram-positive anaerobes do tend to over-decolorize and can look gram-negative. Here is a gram stain of Propionibacterium acnes, and that species name gives you a clue of some of the types of infections. So Propionibacterium acnes is one organism that can cause severe acne. So it gets deep into the skin and the pores and can cause infections in the skin. We're going to move on to the fifth part of our anaerobe lecture.